All right, so next section, here we go. This is it. This is your organic molecules outline. So in front of you, you guys will have kind of your first set of notes. And I say first set because we'll kind of go through it a few times. You should remember this from first year bio, and I keep saying that to you guys, and I know a lot of it may not have stuck with you. This is important stuff. This is kind of the nitty gritty of your cells, of your micro, of what makes up tissues, of what makes up organs, of everything else that kind of makes our body going. Proteins, you'll see carbohydrates, you'll see lipids, which are your fats, and you're going to see nucleic acids, your DNA and RNA, as well as your ATP, which is your energy source that we see through cell respiration. All right, so why did we have to do basic chemistry? And the reason is because we look at carbon here and we break down carbon a lot: protons, electrons, neutrons, valence electrons, how they bond together. And it's really important that you guys were able to understand that particular section because without carbon, we do not have organic. So in order for something to be considered organic, it must contain the element carbon. Are there exceptions to the rules? Sure, CO2, carbon dioxide. It contains carbon. It is not an organic substance. So there's always some kind of exceptions to our rules here. So if you remember carbon on your periodic table, here it is up here. It's got an atomic number of 6, and it's got an atomic mass of 12, 6 protons, 6 electrons, and 6 neutrons. Those protons and neutrons are what are within the nucleus, and that gives you the mass of 12 there. Those 6 electrons are floating in the outside rings. So you have 2 in the first ring because we said that first ring can only hold 2 electrons. And then you've got 4 in the outside ring or the second ring. So first ring is stable, it's got the 2. I throw my other 4 that I have left over there because we have a total of 6 into that second ring. And that means I have 4 valence electrons. I have 4 electrons that are available for bonding. So carbon is super unstable like this. It wants 8 in that outer ring. So it either wants to kind of get 4 electrons from someone, borrow 4, maybe get rid of a 4, something to stabilize that outermost ring. So, as its results ability to share 4 electrons, carbon can form tetrahedral structures and shapes. The way that it kind of bonds with it can kind of grab onto and bond anything, it makes these really cool structures that we're going to end up seeing here. Carbon is able to form covalent bonds. We said that we share electrons with those covalent bonds, and they can covalently bond with other carbon atoms. So you guys are going to see these really giant carbon chains, hydrocarbon chains, especially within our lipids. So the bonds between carbons are going to be strong. The atoms are able to form chains, branch chains, as well as rings. Usually within your sugars or your carbohydrates, you're going to see rings. Within your lipids, you're going to see these long chains. Addition to the change can change activity to it. So when we get to like proteins and other little functional groups, we can actually change the purpose or the function of that particular carbon in terms of bonding and what that structure is going to become. So once again, under D, it says addition to the chain causes a change in activity. Additions can take form in special groups called functional groups. You're going to see them in this video here. Or additions can increase the number of carbons, long hydrocarbon chains. You will see it when we get to lipids, or within the ring, and that's what will kind of create glycoproteins and lipoproteins when we get to our protein section. Bonding is shown by that little line that you guys see there as little symbols, can sometimes be dotted lines. The formula shows the position and arrangement of the bonds around carbon is called a structural formula. So down below, you guys are going to see some of that. So here's my little structural formula. This one here is kind of a ball and stick model. It shows more your tetrahedral structure and the way that it can create this 3D structure because of the way the bonds are arranged around it. When you look at your molecular formula is the same thing as your chemical formula, your empirical formula. It shows the number of each atom in a molecule, how many we have, what elements we have. That's our molecular empirical formula. If you see a single line, just a plain single line, we are sharing a single pair of electrons. That's going to create a single covalent bond. We see them within our sugars. You guys can also create a double bond or a double covalent bond. It means that they are sharing 2 pairs of electrons. So basically 4 electrons are being shared, and mostly between carbons is what we're going to see it. Sometimes you'll see carboxyl groups, a functional group, and we'll see those double bonds there as well. Sometimes you'll see a triple bond. It's a little bit more rare, but what's happening here is that they are sharing 3 pairs of electrons. So 6 electrons total are being shared between those 2 elements in order to stabilize those outermost rings. And that's what bonds do, whether we have a single, a double, or triple bond, or how many electrons we're sharing together, we want to stabilize the outermost ring. We want to fill the outermost ring. So anything first past that first ring can hold up to 8 electrons. But I said you must fill the previous ring before you move on to the next one. So if we're looking at carbon, if you remember carbon, we had 4 electrons in the outermost ring. We need 4 more. So I can put some hydrogens there. Hydrogen's got one of its own. It wants 2 because that first ring is only going to hold 2 electrons. So it can actually share a pair of electrons here. That gives carbon 5. We're sharing another one here. That's 6. We share another one here, 7. We share another one here. That's 8. So we actually have 4 covalent bonds going on with these 4 hydrogens. Hydrogen is happy. It's got one of its own. It's sharing one of carbon's all the way around. Carbon is happy. It's got 4 of its own, and that's sharing 1, 2, 3, 4, and it's got 8 in its outermost ring. So that is a stable structure there. So we just talked a little bit about these carbon chains. So you guys are going to see some things called hydrocarbons along the way. And hydrocarbons are just what they sound like. They consist only of carbons and hydrogens. So long carbon chains. Every place you see a little line, there's a single bond and they're sharing a pair of electrons. So this carbon's sharing an electron with this carbon, it's sharing with this carbon, and they're stabilizing themselves. We have some hydrogens that are also covalently bonded there. When we see this, we can create things called alkanes, alkenes, and alkynes. Now notice the highlighted letters that are there for you guys. Alkanes have an A, alkenes have an E, alkynes have an N. And these are words you may have heard in chemistry class and may have worked with them a little bit differently. But it talks about the carbons and how they have to have 4 bonds around the nucleus. So if we have all single bonds, carbon and hydrogens with all single bonds, it is known as an alkane. So it kind of goes alphabetically: single bonds, double bonds, triple bonds, A, E, Y. If you're looking at alkenes, you have a carbon and hydrogen structure with a double bond. If you're looking at alkynes, you have carbon and hydrogen with a triple bond. So you guys kind of see a little formula there. Okay, so we're talking carbon and hydrogen. So if we go up to our alkanes, we can say, okay, C to the N, H2N plus 2. So if I go through and I look at something like, let's look at methane, which is my next little page here. Methane's got 1 carbon. So C to the 1, so I have 1 carbon. H, 2 times 1 is 2 plus 2 is giving me 4. So I know C1, H4. I know that if I'm dealing with an alkane that has 1 carbon, I can calculate out how many hydrogens. If I have an alkane, single bonds, with 2 carbons, I don't have to sit there and draw it and count out my hydrogens. I can go, oh, okay, CN, so I have 2 of them, H2N, so that would be 2 times 2 because I have 2 carbons there. That would give me 4 plus 2 more, it's going to give me 6. C2H6. Oh, there it is. Can you draw it out? Sure, but when you get up to things like, 5 carbons and above, you're really going to have to start sitting there and drawing things out. Where if you remember the formula, then you guys should be able to calculate out in your head. If I'm going to alkenes, I'm getting rid of the plus 2 here. Remember, we're going to have a double bond someplace, so we're sharing more of those elements together, those electrons together. So that double bond means that we're going to share a pair of electrons. So 2 carbons would give me 4 hydrogens. I actually will break off, and because of that double bond, I don't need as many hydrogens there. When I go to alkynes, same formula as what we have up here. Instead of a plus 2, we're going to subtract the 2 because now we have a triple bond and they're going to be sharing more of those electrons together when there's no place for those extra hydrogens to be. Okay, so as we start going through and creating these double bonds and triple bonds, you guys will see how we need less and less of those hydrogens in order to fill those spots, especially with your lipids. You guys are going to see, if you remember, saturated and unsaturated fats. We'll talk about these hydrocarbon chains with them and how many we can actually fit on there because of the number of bonds that we have. So we have structures like methane, ethane, propane, butane, and pentane. So these are my hydrocarbons. They're just hydrogen and carbons. They're these long chains. And memorize your formulas above for them. Isomers. An isomer is a compound with the same molecular formula and different structural formulas. So far you guys have seen things like an isotope. We talked about them in your basic chemistry. So another little I word for you is going to be an isomer. Same chemical formula, different structural formula. So when we get to like C6H12O6, we're going to talk about isomers of your sugars. Monosaccharides, isomer disaccharides, meaning they all have the same chemical formula. So first year bio, C6H12O6, you're like, oh, that's glucose. It is glucose, but it can also be galactose. And we can kind of draw it out a little bit differently, and it gives you a slightly different sugar, but they all have the same chemical compound. Here in front of you guys have the example of heptane, C7H16. Our formula above, we said C to the N, so I have 7 of them. H to 2N plus 2, so 7 times 2 is 14, plus another 2 gives me 16. So without drawing that out, I know that heptane, which has 7 carbons, is also going to have 16 hydrogens around it. It is all single bonds, which is why you see all these little lines around these structures here representing single bonds. If I put hydrogens on all of them, it would give me exactly 16. Earlier on, we said if you start putting these functional groups or different functional groups, you're going to change the function of your carbon or the structure that we're creating. So these are some of the functional groups that you see here. They can be attached to a carbon skeleton. So your hydrocarbon chains, we can hook some to there. And it's basically going to change some of what we're going to see here. First one, hydroxyl group. It's that OH group that we see there. So we'll actually bond that OH. You'll see this little bond here, and then we bond it to a structure. And you'll see it a lot as we go through a lot of our organic structures. It's water soluble. It's got the elements of water in it. It's created by hydrogen bonds. It's polar, just like your water molecules are. We'll see it a lot with your alcohols and methanols there. Carboxyl group plays an important ending in amino acids, which make up your proteins. You also see it when we get into our lipids. It's going to make part of that fatty acid chain there. So that carboxyl group, you guys will usually see it as COOH. But when we draw it out, here's my carbon. Remember, he's got 4 electrons in the outer ring. He needs 4 more bonds. So there's one of his own, 2, 3, 4. So he needs to bond to 4 things. Well, he's going to bond to another carbon on this side. So there's 5. He is going to bond to the oxygen here. That's 6. This oxygen is then bonded to that hydrogen. And then he's going to double bond to oxygen. Oxygen's got 6 electrons in the outer ring. It needs 2 more, so it's going to share 2 with carbon. So carbon's got 4 of its own. 5, 6, 7, 8. It is stable. That carboxyl ending is a weak acid. It also is going to have a negative charge. So when we get to our protein lab and we start splitting apart your amino acids, you guys will see some of this material as well. Your amino group is the other end of your amino acids, the NH2 group. So there's nitrogen and it's bonded with my hydrogens as you see there. And it'll have another bond coming off this way. Usually it's going to be bonded to a carbon is what we'll end up seeing. This amino group has a positive charge to it. It's a weak base and it's going to help to make up individual amino acids, which if you put amino acids together, they are your building blocks of proteins. An aldehyde group. So here's carbon. It's double bonded to oxygen. It's got 1 hydrogen. And on this side, we're going to be bonded to more carbons along the way. We'll see some of these aldehydes in some sugars. They are water soluble, especially if they're sugars, and they're also polar structures. Ketones, they look pretty similar to your aldehydes, but we're losing a hydrogen and we'll have 2 separate bonds over here, possibly 2 separate carbons over here, or other structures. Same thing as your aldehydes, they have the same biological activity. They are water soluble, they are polar molecules, and we find them in sugars. So ketones and aldehydes are within your sugars. Phosphate, PO4. So here's my phosphate, here's my oxygens all the way around there. This is a high energy bond. We'll see it a lot when we get to our nucleic acids within our DNA, within our RNA, and also within the ATP, which is the energy molecule within our body. It's a hydrogen ion donor, so we see it a lot within acids, because remember, acids, we're checking for those hydrogen ions in solution. And you guys will see some of these a little bit later on. We'll talk about them more as we break down our different organic molecules, the 4 that we're going to see here in a moment. Energy factor. So we talk about bonds and we talk about how these bonds can store energy and how they can release energy. So remember that covalent bonds involved in sharing are very strong. They're very stable as long as the number of electrons there are in order to stabilize that particular element. The atoms and molecules are also in motion. So it's not like, hey, we're bonded and everybody's just kind of stuck in one place. They're constantly moving, especially if they're sharing electrons. They're moving back and forth between those energy rings, between those 2 different elements. They're vibrating, they're rotating. If the energy of motion Energy in motion is your kinetic energy becomes great enough, the bond is going to break. When that bond breaks, that's when we release energy. Okay, usually this is going to be in the form of heat. We see that within food chains and food webs. So, the cow comes along and eats the grass that got its energy from the sun. 10% rule. We're losing 10% of that energy for every trophic level. Reason is because when that bond gets broken and we're transferring energy to the next organism, some of it's going to get lost as heat. This energy is measured in kilocalories per mole. Once again, that's probably first your chemistry stuff that you guys talked about. What you need to remember, when bonds are broken, chemical reactions occur which cause the production of new substances. When bonds are broken, you are releasing energy. When we form bonds, you are storing energy, and that's that potential energy. We're going to talk about catabolism and anabolism once again. Catabolic reactions, anabolic reactions, dehydration, synthesis, hydrolysis, building up, breaking down. So those were words that we talked about with those chapter 1 notes. So don't forget those particular words either. So if we think about it, we have our reactants. We can break those bonds. These little arrows that you see means yields. It's going to give us a product plus some energy release. And a lot of times that energy is going to get released as heat. These reactions that we're going to see here are going to be controlled by enzymes. Enzymes are a special type of protein, actually. So we'll really talk about them at the end of this section. But enzymes can also be known as catalysts. Anything that can speed up a reaction without being used up over, or can be used over and over and over again, unless something denatures it. And we'll talk about that denaturing more when we get to gel electrophoresis. And we'll talk about the buffer and how that buffer has to have a certain pH so that our molecules do not denature or get destroyed along the way. Okay, so this is just the first little part of it. I'm going to kind of break these notes up into little sections for you guys. So next we'll look at our carbohydrates, then we'll look at some lipids, and we'll kind of end with proteins and nucleic acids. Then we'll come back and we'll look at it in diagram form as well. And we'll talk more about these terms that you guys have: catabolism, anabolism, dehydration, hydrolysis. They're going to be the same words in each one type of your 4 organic macromolecules. So I can build up and break down carbs. I can build up and break down lipids, build up and break down proteins. The same way: dehydration, synthesis, hydrolysis, anabolic, catabolic, making bonds, breaking bonds, releasing energy, storing energy. Everything's going to stay the same. Please keep that in mind. Okay, so we'll finish this up, or we'll start with our carbohydrates next time. As we go through, still again, keep yourself a little list. Hey, I'm not quite sure about this. Let me ask her. I'm not quite sure. This information is going to build upon itself. So if there's something you don't understand with bonds, or the way that those electrons are able to go through and create ionic bonds, covalent bonds, share electrons, give or take electrons, create anions, create cations, it's going to keep building. So if you don't know A, B, and C, you're definitely not going to know the rest of it. Okay, so ask those questions along the way. Thanks.